content that we are producing every day. Secondly, well, I'd like you all please to keep muted during the presentations, but when it comes to questions after, feel free, if you're called upon, to unmute yourself and ask them yourself. Otherwise, I'm quite happy to read out a question. And of course, if you do have any questions that pop up during the course of the presentations, feel free to use the chat function in order to lodge your questions, or use the raise hand function under manage participants and say hello. So without any further ado, I think, Mary, I would like you, if possible, take it away.
Okay, thank you, Brinley, for hosting. Let me share my screen, press play. Okay, thank you everyone for coming today. I think it's a really cool thing that the STEM Village is hosting this venue for LGBTQIA+ scientists to share their work. And so I'm here to talk to you about insect wings, specifically the hydraulics inside of insect wings, and kind of poke your curiosity on why you should care. So I work at Virginia Tech in Blacksburg, Virginia. That's West Virginia. And a land acknowledgement recognizes the native peoples who were on that land before we all came. And so Virginia Tech is on Tutelo-Monacan Nation land. And to do a land acknowledgement is just to recognize their elders, past, present, and emerging. Insect wings, they're dynamic, they're flexible, they're flappy, they're twisty, and it requires a huge amount of hemolymph and circulation in order to keep these insect wings flexible. Insect wings inspire invention at so many different levels, from micro aerial vehicles, thinking about, can we build those tiny flapping robots, to larger inflatable wings that may be dropped from another aircraft, to, again, drone surveillance or emergency services. I love this example, the morpho butterfly, because it's the nanostructure of the butterfly wing scales are actually perfect for building like nanophysical materials. So thinking really tiny scale on a whole other side of the spectrum, not even robotics. And so I'm interested in wings. And what are they exactly? Well, I flipped the phylogeny earlier to take a picture of myself on Zoom, because Zoom flips all the text. So this is just the insect phylogeny with all the order names flipped. And so across our insect phylogeny, we've got wings that have really sparse venation and dense venation. And inside of each of these wings is a really important protein called resilin. And resilin is this flexible super protein that needs to be hydrated in order to function. And so I'm showing you a cross section of a wing vein right now where everything in bluish purple is this resilin protein. So a wing is first structurally comprised of resilin and insect cuticle, and there's spacing inside of it. This resilin is really important. If the wing was flapping all around, it would probably break really easily without the resilin. Wings are also microchips. They're covered in sensory spines. Some of these are gustatory sensors, so they're actually tasting chemicals that may flow over the wing, or they're mechanosensors, so that as they bend and deflect, they literally bend the neurons, sending information back to the insect about the flight pattern. Or there can be tympanal organs, so insects that make or receive sound, but on the wing. And so all this to say that what's in a wing is very important about what the wing supports. So in order to make sure our flexible proteins are functioning, our mechanosensors and sensory structures are functioning, we need a suite of things inside of the wing. And that's a supply of hemolymph, so insect blood, tracheal supply, so that's oxygen, and again, these supply of neurons that branch throughout the wing. And if we look at just like another cross section of that wing, you see that wing cuticle is layered. And again, we have that resolin there. So it's a suite of factors that are living. So I'm showing you one of my movies. And this is a movie of fluorescent particles moving with the hemolymph current in the wing, in the veins. So I pause because I find this extremely beautiful. And I want to just give you a taste of what it actually looks like in real time in the wing of hemolymph circulating. So we need hemolymph in the wing for a lot of reasons. Hydration is really important, and nutrients. Oh, I'm showing you, or I'll get to that movie in a second. The presence of resolin, which is actually found throughout the insect body. And then our sensory structures, such as tympanal organs or sensory spines. There's also another key component that I'll get to into the talk about the hydraulic expansion of wings. What I think is really interesting is this presents a series of questions that I'm just starting to answer in the Salcedo research program, and thinking about our predatory insects, so our dragonflies that eat mosquitoes and flies and other dragonflies. Do they need a higher supply of hemolymph in the wing because they're performing these aerial moves that are really dynamic? Or our migratory monarchs, thinking about the distances that they go, do they have to have a specialized circulatory system to make sure that their wings are maintaining their flexibility? Or does that flexibility just decrease as they migrate and age? And we do know that age decreases wing flexibility. Or what about insects that may feed at cooler times of day? Are the wings requiring a higher flow of circulation? Why do we care about that generally? Because without wings, our insects couldn't perform all the functions that they need to do, such as pollination. And so I focus on not only the physiology of the wing, but the biomechanics of the flow and how the wing deforms in flight. And then I'm really getting into this kind of applied agriculture and thinking about where do pesticides go in insects based on their circulatory system. And so all of these systems are coupled and they all affect each other. And that's where I'm trying to like focus my upcoming questions on. And so I can look at this in several different ways. And I'm grateful that technology, microscopes have come so far, because one, we can look at the inner structure of wing veins using high-powered synchrotron X-ray. We can look at the circulatory flow from what I just showed you with injecting fluorescent particles that move along with the flow in the insect wing. And we can sample pesticides in insects very specifically and learn about where they accumulate. So if you're unfamiliar with insects, insects have an open circulatory system, meaning that blood flow is just everywhere all over the insect body. They do have a long dorsal heart. It's a tubular heart that pumps flow from the posterior end to the anterior end. And then they can have accessory hearts. So you'll see that there's a little antenna heart at the head, and those large pink V's actually mark wing hearts that are found in the thorax, and they pump flow into the wings because their heart is not enough to just extend flow into its appendages. In contrast, we have a respiratory system that's closed. So this is a network of tubes. And in the thorax, there are openings to intake air. And then there are openings that are called spiracles in the abdomen to expel air. And so this works to deliver oxygen directly to the tissue. And so that's why we find trachea inside of the wing veins as well to deliver oxygen to live tissue deep inside the wing. And so I'm here to ask, across this insect phylogeny, and we know that wing circulation is important for wing flexibility, wing sensory structures, do we really know much about it? And the truth is we don't. And we know kind of a little bit about mosquito circulation and kind of a little bit about butterfly circulation. But a great question we can ask just to start is, does pattern affect circulation? And so I turned to the North American locust, and that's a species that I can get through the U.S. Department of Agriculture, and I can take as many as I want. They often ship them to me in the hundreds. And I can look at circulation throughout their bodies and their wings. In respect to like where we know they fit in to the knowledge base of circulation, we really only have like 3 species. We have mosquitoes, the Atlas moth, which has a very large wingspan, and now we have this grasshopper. So how do I do this? I start with live imaging. The grasshopper is gently restrained, and I do high speed, so just over our general 30 frames per second. So I looked at 9 grasshoppers. The grasshopper was literally restrained with like clay or Play-Doh, and then the wings were just pinned in a 2D format. I'm able, because of that open system in the insect, to inject particles in the abdomen of the insect. So these are neutrally buoyant particles that just get sucked up into the entire body, and those little wing hearts actually just push them into the wing. Then I can track those particles, because we have our 2D trajectories, and find out flow velocities all throughout the wing. Because I want to get into a bunch of different topics, I won't get into the full data of what that shows about how pattern affects wings, but I can show you what it looks like. And so what you're looking at here, you can see that little blue rectangle in the bottom right. We're looking at the forewing of this grasshopper, and you're seeing those bright particles move along with the current of blood pulsing into the wing. Because we're right at the wing base, we're very close to our pulsing hearts. And so you can actually see that kind of pulsatile nature in the flow. What I also want you to look at is this kind of like gray matter moving along with the flow, and like little particles. Those are blood cells. So blood cells are actually being pushed and pulsed into the wing. And you can see that my particles are a pretty good proxy for blood flow. If you look closer, you can see little tiny white branches, and those are nerve branches. So the nerve hairs are out of focus in this angle, but those are actually branching to sensory structures. And so back to the title of this slide, it's called Pulsatile and Leaky. So that's referring to the flow. So you're familiar with what a wing vein looks like. I showed you this phylogeny. There's all of these veins. The veins themselves aren't necessarily closed structures, so they're not just tubes. And in this specific region of the forewing, I call it leaky, because we actually have this section where flow is just kind of moving in the wing membrane itself. And so this is kind of a unique thing that entomologists are starting to recognize, because it's not really been known before or shown before. So this is pretty cool stuff, showing that there's a lot of live tissue in the wing because there's a hemolymph supply, and this hemolymph supply really affects wing flexibility. So across this wing, and you'll have to wait for the paper, we see 4 modes of flow. So I have movies on movies on movies, but I want to show you that there's a lot of different flow types across this very dense wing, where we have pulsatile flow, meaning it's really back and forth. We have leaky flow, and that occurs in a couple different regions in the wing, not just in the wing base, but also towards the wing tip. We have lattice flow, so our bidirectional flow, where a flow is actually moving at itself. And then we have that leaky flow right there, like 3/4 of the way down the wing, where flow is actively moving into membrane, which is pretty unique. So I gave you a teaser about like what's inside of an adult insect wing, and now I want to go into why wing hydraulics in general, and why does an insect even need to depend on circulation in the wing. You're like, okay, there's circulation in the adult wing, but circulation plays a major effect at development. So you might be familiar with from classrooms, just like, oh, you have your monarch, caterpillar, pupates, then all of a sudden you have this beautiful monarch emerging. Well, this wing expansion phase that happens at emergence, where the wing is then unfurled and expanded to normal, ready to go flapping size, is a bottleneck for the insect. And if it's not performed correctly, the insect's viability is just nothing. So this poor little moth that I found on the right-hand side has no chance of a life afterwards. It cannot pollinate, it cannot feed. And so it's really important that those hydraulics happen in time and that the hemolymph is pumped into the wings. And so again, back to our North American grasshopper, these insects actually form an instars. So outside of the egg pops a little version of the adult, and then that adult grows and goes through growth stages. And at about the third growth stage, you see the development of wing pads. And so these are pockets where the wing forms. And what's really beautiful about this is it almost forms like an origami structure that's just like waiting to unfurl. And so when the insect goes through that expansion phase, it literally, like a sword out of a sheath, it pulls this wing out of the wing pad and then starts an expansion phase. So I'm going to show you what that looks like. So that's super fast, where you just saw the wing unfurl and then get folded. This happens in about 40 minutes. So the right-hand side is a little bit longer of a time lapse. It should start to load. Oh, I don't know why it's freezing so much. At least you're seeing the left-hand side. Okay. I don't know if it's freezing for you. It's freezing a little bit for me. But hopefully you can see that there's some pumping of the insect body that's happening as well. So the insect pulls out its wings, starts to unfurl them. There's active pumping in the thorax and the abdomen to push fluid into those wings and expand them to full size. Even that last little bit of folding from the hind legs is critical to the wing actually having its final shape. So I love this expansion phase because I'm so curious about how the hydraulics are actually unfolding this tiny, tiny wing. And I'm gonna now jump to like what we know of what's inside of a wing. So I showed you a cross section earlier. There's space for hemolymph, there's space for trachea and nerves, but do we actually know the internal structures of a wing, and why should we care? Well, we care about the hydraulics, we care about this hydration. We also care about the technology that we could build from this, thinking about microfluidic devices and other unfolding structures. But it's really hard to see that detail. So often people look at it through SEM, scanning electron microscopy, where you take slices of the wing at discrete sections, and then you're able to look at the beautifully detailed structure. But if we want to know what the continuous structure of a wing vein looks like, we have to do something a little more complicated. And so myself and my PI, Jake Soha, went to Argonne National Laboratory, which is outside of Chicago. And we went to, this is a synchrotron beam. So there are 36 hutches around this beam that focus an X-ray into doing a bunch of different things. So you can look at muscle structure, you can look at structures of metals. And so we look at the inside of insect wings. We use it for high-powered micro-CT or microcomputed tomography. And basically what this looks like is we take our sample, we put it in line with the beam. So you can see our wing is actually curled inside of a tube. And we take our sample to our hutch, and we try to fit it in. And then there's a series of switches to get out of the hutch to make sure you don't actually close your lab mate inside. And then you're able to take really high resolution photos. And so that's just the detector and our sample. And what's really cool about this and pretty unique, so I'm showing you data that's actually brand new, and it's things we've never seen before inside of insect wings, or that we have a record of, like what's happening inside of this continuous small structure. We can see sensory spines that will come out when we do the full 3D reconstruction. That's marked in green. We can see our trachea space, so where all of the air tubes are. And then we can see where the hemolymph space is. So these insect wings were freshly cut off of sacrificed grasshoppers and then sealed so that they maintained their hydration. So we can see this incredible amount of detail. And so if we were to just look at a slice of the wing, so the forewing here is on our right-hand side, we're to look just at a stack of images that I've turned into a movie. You can see that the inside of the wing is actually a pretty dynamic structure. We have air spaces, tracheal tubes that are moving back and forth, sensory spines occurring in different areas. And I was boggled to look at these slices of wings and see that the internal diameter is nothing what I expected. And so this is actually really important for any insect biomechanist that studies flight, because now our understanding of how the wing deforms and bends during flight could be very much informed by the beam structure, the vein structure that we're now discovering. And so this is wrapping up my talk. I think it's really important I see this question asked a lot on Twitter, is like, what are good qualities of scientists, especially in like grabbing students, and I'm constantly trying to find awesome students and thinking about key qualities in a scientist. And that's definitely, for me, curiosity, resilience, so you keep going even when something goes wrong, and creativity. And I just like to discuss these a little bit, because I think all of these can be learned skills as scientists. And I think we don't often go through the process of being like, yeah, you learn how to be independent. You learn how to ask good questions, and you learn how to troubleshoot. And so I've just been thinking about that more and more, getting into the stage of thinking about my own lab and thinking about how I would train scientists. And I find it's a really nice thing to just let simmer in my mind. So thought I'd share it with you. And then I'd like to thank my current lab. We study flying snakes and insect circulation. So I've become a little obsessed with snakes now. And these are my former advisors who helped with my circulatory work. And I will take any questions, because we have time before Glenn's talk.
Thank you so much for that, Mary. That was really fascinating. I had no idea that there's anything within a wing other than just like a thin film membrane with some spines to hold it up.
Yeah. There's a lot to it here. I'll actually just keep it on play. Or should I stop sharing my screen, or trying to see the chat?
Carry on sharing your screen for the time being. We've got a question in from Adam. He asks, during the wing expansion, I assume there must be some blood pressure change. How do insects deal with that?
There's a lot of blood pressure change. So actually, I didn't show you the upside down phase before. Insect, it's emerging. All of these insects actually have an upside down phase where they're upside down and they're breathing in air to stretch out their exoskeleton. Recall, insects don't breathe through their mouths. They do that purely to create a big pressure change and stretch out their exoskeleton. That pressure change in the wing is really interesting, because the wing actually goes viscoelastic as it starts to stiffen. So the materials are actually changing properties in real time, that the insect is changing internal pressures. So we have this viscoelastic, we have this stiffening part of the wing, and then we get like this kind of traveling pressure gradient that unfurls the wing. So how do they deal with it personally? Well, I think very well. But I think that it's because their body is changing all throughout that time. And then even once they emerge completely, the next 24 hours is spent stiffening their entire exoskeleton. So I guess you could say, that was a long answer to say, they soften their bodies to deal with the high pressures.
Fantastic. I hope that answers your question, Adam. Vinko, your hand is up. Would you like to turn on your mic and have an ask?
Hi again, Mary. That was a really, really fascinating talk, and I learned a lot. As an ecologist, I don't know very much about what goes on inside of bugs, so that was super fascinating. So I'm curious about the efficiency of these systems, because in vertebrates, you know, we've got a closed, well, most vertebrates, we've got a closed circulatory system, so blood is always flowing directionally. But what's going on in the wings or the rest of the insect body where there's no, I guess, not necessarily directional flow or uptake to go back closer to the trachea?
Absolutely. So efficiency is a really, really good question, because perhaps it's not efficient at all. And so there are circulatory patterns inside the wings. And I didn't really get into that, but there are different types of circulatory patterns too. So generally there's like an out current, it goes to the wingtip, and then it returns back to the wing body. So you see like this circuit. Whether it's efficient, I don't know. We're still discovering, like I looked and measured speeds of flow. And now that we know internal diameters, I think we can actually get an efficiency measure because I don't know that it is happening efficiently. And then we didn't even touch on what happens during flapping flight. So the wing is going back and forth like crazy. I don't know how that changes flow in the wing, because it's extremely hard to visualize. If like a bee wing is flapping at 200 flaps per second, or 200 hertz, and you're trying to like look at flow movement, I'm not sure how that changes the dynamics of it. So efficiency is a really a good puzzle.
That's super cool. Thank you.
Yeah.
Mary, I had one more question for you, but we're running tight on time.
Okay,
So
I can be quick.
can I ask you to put your email address or Twitter handle or something that you're happy to take questions on into the chat, and then we can copy and paste it and find it afterwards?
Yes.
Fantastic. Thank you so much. In the meantime, Glenn, do you want to get your slides ready and we can transition over?
Yes, give me a second once I figure out how things work on Zoom. I will apologize. I do live in a very kind of family-oriented area, so everything